这么说
3岁小儿，大智难成。
猜忌，
孤侯少年困顿，却能够立即而存，为什么？这和大娘娘都做错了事。
Billy told me what you said. She told Jared too. Last night, and now he's gone. What do you mean he's gone? He's not gonna find him.
I can't believe my only daughter is about to get married. But I am always.
Okay.
下了大树所门进。2右床靠手门开大。张图看一下。
Welcome to week 6. So as you know, unable to have a live tutorial, so I've recorded these answers for you. Of course, for these questions and for any of the tutorials, if you've got additional questions, please do reach out. Some of these content, particularly today and the next week's lecture, are complicated, so make sure you reach out and ask for help. So first one, explain the fate of pyruvate under anaerobic conditions. How does the process differ in the presence of oxygen, and what is the role of acetyl-CoA in cellular respiration? So 3 parts, so similar to the style of question you might see in the end of semester exam. Make sure you read them, make sure you answer them individually. So as we see here, pyruvate produced by glycolysis is metabolized to ethanol or lactate in the absence of oxygen. And this is obviously dependent on the individual organism. So ethanol for yeast and bacteria, and lactate for humans. So in the presence of oxygen, the pyruvate is converted to acetyl coenzyme A or acetyl-CoA. This then enters the citric acid cycle, which we saw previously. This is the key metabolite that enters the citric acid cycle. And so when it does so, it's metabolized into carbon dioxide and water. Which, remember, the CO2 is what we exhale. this process generates energy in the form of ATP, and this is really where oxidative phosphorylation comes into play, because we're feeding these molecules into the oxidative phosphorylation pathway. And this process is called cellular respiration because of the presence and the use of the oxygen. So that's what the respiration is referring to. Now, we have covered these points previously, but I don't think it's bad to have another repeat because it's important to understand these roles in this particular context. So if we now move on to question 2, describe the role of the pyruvate dehydrogenase complex in cellular respiration. So we spoke about this enzyme and really the 3 enzymes that are involved, so you know that this is really quite important. What's the significance of the irreversible conversion of pyruvate to acetyl-CoA? So remember, pyruvate is where we're starting, and we get it converted to acetyl-CoA. Once you go on this direction, as it says, this is irreversible. This is a committed process. So remember, glycolysis occurs within the cytoplasm, whereas the citric acid cycle takes place in the mitochondrial matrix. There are different locations. That's important because it helps to segregate or separate the substrates to different locations and facilitate the location of the enzymes so that you get the correct processing rather than all having them in one location. Now, for this separation to occur, pyruvate has to be first transported into the mitochondria because, as we've said, we've got cytoplasmic versus the matrix, so we need to bring it in. That's one step, and we'll see this later when we come to talk about fatty acid metabolism. You'll see a similar thing where fatty acids have to be activated before they can be metabolized, and that activation requires this transportation. The pyruvate is then oxidatively decarboxylated. So you've got an oxidative reaction, so again redox, decarboxylation is telling you the removal of CO2. And the enzyme that does this is pyruvate dehydrogenase. And it's not one enzyme, it's actually a complex of 3 enzymes that each have a different prosthetic group. But the key point is, this is a massive complex. And what it does is, as you can see in this structure down the bottom, pyruvate plus CoA plus NAD+, this is part of the oxidative and reduction process, gives rise to your acetyl-CoA with CO2, NADH, and a proton. And so all we've done is convert the pyruvate into acetyl-CoA. And as it says here, irreversible conversion of pyruvate to acetyl-CoA is the link between glycolysis and the citric acid cycle. When you're at glycolysis, if you're at this pyruvate stage, you know that it can go back and it can be used in different pathways. Going to acetyl-CoA means that it now has to go into the citric acid cycle and ultimately give rise to ATP. So this commits carbohydrate-derived carbon atoms. That's what pyruvate is. Pyruvate is carbohydrate-derived carbon atoms to oxidation by the citric acid cycle. Alternatively, and we really won't go into this in a lot of detail, acetyl-CoA can be used to make fatty acids because fatty acids are essentially the addition of acetyl-CoA units. And what we'll see later really is from the other end, where we're breaking down fatty acids.
水又放齐了吗？我去封门，哥们儿，我去封门。封正门，封门没有用啊，小 K 封门没用啊。图片吗？吹图片，哥们儿。
you'll see that we will generate huge amounts of acetyl-CoA, and that in turn gives rise to a lot of ATP. So, what are the steps? Conversion of pyruvate to acetyl-CoA involves decarboxylation, we said that here, oxidation, said that here, and transfer of acetyl-CoA to coenzyme A, and we see that here. Remember that the pyruvate dehydrogenase complex was 3 enzymes, and notice we've got 1, 2, and 3 steps. So each step is performed by the pyruvate dehydrogenase complex. So you've got all of these enzymes together participating in this sequential reaction. And because this is the first step, this is going to be the important molecule or the important enzymatic group. And again, I've highlighted that to you multiple times. So, if that is so important, how is it regulated? And again, this is the same process that we spoke about before. I'll highlight to you the key enzyme, and then we talk about how that key enzyme is regulated. So how does the phosphorylation of pyruvate dehydrogenase complex serve as a key regulatory mechanism? What factors inhibit and activate the pyruvate dehydrogenase complex? So, the pyruvate dehydrogenase PDH complex is regulated by multiple allosteric and covalent modifications, and we'll see this later. So the allosteric can be the inhibition and activation, and the covalent modification refers to the phosphorylation. Now remember, animals, so us, we can't convert acetyl-CoA back to glucose. We saw that before when we were talking about in the context of pyruvate. So the acetyl-CoA must be converted into energy. As soon as it's made, it goes into energy, citric acid cycle, or synthesis of fatty acids. But what we're talking about in this lecture is the citric acid cycle. So our focus is going to be on the movement of acetyl-CoA into the citric acid to make ATP, ultimately. So, how do we inhibit this complex? Well, if you've got high levels of acetyl-CoA, that's already suggestive that there's a lot going into the pathway that's going to turn off the PDH complex, because you don't need to feed it in if there's already a lot of acetyl-CoA. That's going to be wasteful. So what would occur? This acetyl-CoA would be turned into fatty acids and be diverted into the synthetic process. And similarly, NADH will inhibit the dehydrogenase. Now, if we go back to the previous example up here, notice the byproduct was NADH. Just like we said, if you've got a lot of acetyl-CoA, if you've got a lot of NADH, this is going to be allosterically modifying the PDH complex. Turning it off. If you've got you've got enough substance.
切了。动不了吗？没人看齐呀。
In the cell to give rise to those byproducts. So there's no need to make them again. So you turn off the enzyme complex. Remember, we're trying to get back to a homeostatic level at all times. So phosphorylation turns off the activity, and you know that the opposite of a phosphorylation event is a dephosphorylation event, and this occurs through an enzyme called a phosphatase. So you can turn it on and off. You can think of it like a switch, and phosphorylation is a very, very common mechanism of regulating activity and pathways of enzymes, and we'll talk about this in depth in the signal transduction lecture. This is the diagram that was in your notes, and this is showing you again those two different states. So the high energy charges where you've got lots of NADH, acetyl-CoA, or even ATP, even though we didn't mention it. But you can see that these all negatively go back and inhibit the pyruvate dehydrogenase complex because they're already present at a high level. There's already a high energy charge, already that potential. So they need to come back and block it. When these are low, pyruvate or Na can stimulate, ADP can stimulate, because remember, pyruvate needs to be converted through the PDH complex to give rise to acetyl-CoA. ADP needs to be converted into ATP. So if you've got more ADP, then there's the drive for it to be shifted towards ATP. Don't have a cycle. So remember, there were two stages. Stage 1, stage 2. That's the first thing. We'll get to these steps in a moment. So remember, acetyl-CoA was a 2-carbon molecule. It condenses, and I've put it in brackets just to remind you, whenever we talk about condensation, it's the same thing as addition. So we add it to oxaloacetate, which is a 4-carbon molecule. And hence, 2 plus 4 gives rise to a 6-carbon product, which is citrate. Remember, this is the citric acid cycle. Citrate, citric. You can see the linkage there. So citrate releases 2 molecules of CO2, and importantly, the electrons, the high energy electrons. And we use the electrons to drive, well, the electrons get transferred to NAD, NADH, FAD, and it is the carrier of those electrons that are then used to make a proton gradient, which ultimately gives rise to ATP synthesis. But again, that's a future lecture. Now, stage 2, 4-carbon compound remains. This is further oxidized and we regenerate oxaloacetate. So remember, we're starting off by acetyl-CoA combining with oxaloacetate, and we're regenerating the oxaloacetate. So here, it's never being lost. That's important because remember, this is a cycle. So for this to continue, we must have this regeneration. And as I mentioned up here, citric acid cycle captures electrons to form NADH and FADH2. These are the 2 cofactors that are really, really important because this is where they carry that electron. And these molecules yield 9 ATP molecules through oxidative phosphorylation. Now, in terms of the steps, but remember, we started off with the acetyl-CoA, oxaloacetate, giving rise to citrate, and we said that right here. Then it is the following steps. So citrate goes to isocitrate. Isocitrate goes to alpha-ketoglutarate, a molecule we have seen often, and we'll see again, even in the context of amino acid metabolism in next week's tutorial. And through this process, we generate a molecule of NADH. So we've got electron transfer. Ketoglutarate gets converted to succinyl-CoA, another electron transfer. Succinyl-CoA goes to succinate, we get a molecule of ATP. So you can see there's not a lot of ATP, but some come through. This is not the bulk of ATP generation, but it's a nice output. Succinate goes to fumarate, and this is now where we get FADH2. How do you remember that it's FADH, not NADH? well, you've got an F. So FAD, easy way to remember, you know that the others are going to be NADH. So the only time that we're making NADH. Furamate goes to malate, malate goes to oxaloacetate, and we generate yet another NADH molecule. So you can see a lot of outputs got more NADH than FADH2 and just one molecule of ATP. I haven't included the names of the enzymes. I think that's, if you know them, that's not a bad thing, but probably that's more than you're required to know. Now, last one. Explain how isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase control the citric acid cycle. These are the enzymes. You can see ASE here. These are going to be involved here. Isocitrate dehydrogenase. Well, isocitrate is here, goes to alpha-ketoglutarate. Isocitrate and alpha-ketoglutarate are those two key molecules. So remember, pyruvate is not the only source of acetyl-CoA. When we degrade fatty acids, and the process is called beta oxidation, there's huge amounts of acetyl-CoA. Because remember, we can synthesize fatty acids from acetyl-CoA, so therefore the reverse is going to generate a lot of acetyl-CoA units. So therefore the primary control points of the citric acid cycle are, not surprisingly, these two enzymes, isocitrate and alpha-ketoglutarate dehydrogenase. These are both allosterically controlled enzymes. You can start to see now, we're seeing allosteric control time and time again. And what this means is that byproducts can turn these off or regulate their activity. So if we look at isocitrate dehydrogenase, it's stimulated by ADP. Again, similar to the logic we said before, if you've got a lot of ADP, you want to convert it to ATP, so we want to stimulate the flow through. And this indicates the need for more energy in the form of ATP. Conversely, when you've got a lot of NADH, which is the result of the end result of that reaction, it will inhibit because you've already got a lot of that product that has been generated. So you can see you've got this balance. And notice you have one side that stimulates, you have one side that inhibits. If you've got a control point, you need to be able to go on the forward and the reverse reaction at those points. Otherwise, you're only going to be able to either stimulate or inhibit. You can't just go in one direction. You need to have two-direction control. And the other enzyme was the alpha-ketoglutarate dehydrogenase. It's inhibited by its products, so succinyl-CoA and NADH, as well as high levels of ATP. And again, it makes sense. The products are there, they can go back and inhibit the enzyme. So again, try to understand the logic. Process, because this is going to help you to understand the process. But before I stop here, and I'll come back in the next part of this recording, remember, because this is not live, you don't have the opportunity in this case to ask all the questions that you to part 2. So part 2 of this recording, we're moving on to the electron transport chain. And so here, we're talking about the citric acid cycle again and the link to the electron transport chain. So how does the citric acid cycle feed into the electron transport chain? Are there any differences in energy? We're talking about ATP here in the products generated by the citric acid cycle, and if so, what? So, in the citric acid cycle, we saw acetyl-CoA being oxidized, we form ATP, just one, multiple molecules of NADH, and one molecule of FADH2. This is not where the bulk of the ATP is generated. We get some, but it's minimal in comparison to what we'll see later. So, in the electron transport chain, this is the final stage of cellular respiration. All that we've done to this previous step is just create these intermediates that feed into the electron transport chain. And this is where the ATP is going to be generated. Remember, NADH and FADH2 are electron carriers. And by doing so, those electrons are used to generate a proton gradient. And the gradient is between the inner mitochondrial membrane and the outer membrane. And it's this difference in protons, this gradient, which drives ATP synthesis through the ATP synthase enzyme. Now, oxidative phosphorylation, or OXPHOS, very short, oxidative phosphorylation generates 26 of the 30 molecules of ATP from the complete oxidation of one molecule of glucose. So you can see, yes, there's some ATP that's generated elsewhere, but the vast majority, 26 out of 30, is through oxidative phosphorylation. Now, when we look at the energy differences, we know that there are differences between the amount of energy, ATP, that can be generated from NADH and FADH2. And this is because they differ in their redox potential. So the number of ATP molecules generated per electron carrier, it is higher for NADH than it is for FADH2. So NADH generates more ATP, 2.5 ATP molecules, whereas FADH will generate 1.5 ATP molecules. And again, we'll see this when we come to look at Now, the next question was talking about the role of the iron sulfur clusters. So the iron sulfur clusters are crucial electron carriers. Remember up here, we've just spoken about FADH2 and NADH, saying that they are electron carriers. They have to transfer their electrons. Where do they transfer them to? To these clusters. So this is like pass the parcel of what we're passing are electrons. So these are crucial electron carriers. They facilitate the flow of electrons through the respiratory chain, because you know that there are multiple proteins in the electron transport chain and they're getting passed from one layer to the next. Those clusters are embedded within the protein complexes. So these are part of those protein complexes. And so they play that fundamental role in transferring of electrons from donors to acceptors. So remember, if we're flowing electrons through, one molecule has to donate it, the next molecule that takes it has to be the acceptor. Iron sulfur complex facilitates this process. Now, we've got multiple complexes, but remember, complexes 1, 2, and 3, this is where you see the iron sulfur clusters. So the first one was NADH dehydrogenase. It's involved in the oxidation of NADH to NAD+, and we transfer the electrons to ubiquinone with coenzyme Q. We can tell that we've transferred that electron because we've lost the proton here. Complex 2 was succinate dehydrogenase. Notice dehydrogenase, we're stripping off the proton. Here we've got the cluster participating in the oxidation of succinate to fumarate. And this is during the citric acid cycle. We saw that above. And the last one is cytochrome B complex. Here we've got the clusters mediating the transfer of electrons from ubiquinol to cytochrome C. And again, this is another electron carrier in the electron transport chain. And remember, transfer of electron drives the pumping of protons across the inner mitochondrial membrane. It is this difference in proton density, or the gradient, which is used by the cell to turn the ATP synthase enzyme, and it is the turning of the ATP synthase which takes phosphate, joins it to ADP, and in turn creates proton motive force. Is all talking about the proton gradient and the ability of the difference in protein concentrations to be used like a tiny battery. So, electron transport chain pumps protons from the matrix to the intermembrane space of the mitochondria, generating a proton gradient. You've got more on one side than you do on the other. The hypothesis for this is called the chemiosmotic hypothesis. And what this says, and we know it's true, is that electron transport and ATP synthesis are coupled by this proton gradient across that inner mitochondrial membrane. We're linking movement of electrons and ATP synthesis to its proton gradient. Now remember, whenever you have a gradient, the system will want to reach equilibrium, wants to go back to its normal state. So the protons will flow back into the matrix to give you this equalization or this homeostatic event. And is that coupled to difference in protein concentrations to be used like a tiny battery. So, electron transport chain pumps protons from the matrix to the intermembrane space of the mitochondria, generating a proton gradient. You've got more on one side than you do on the other. The hypothesis for this is called the chemiosmotic hypothesis. And what this says, and we're not straight, is that electron transport and ATP synthesis are coupled by this proton gradient across that inner mitochondrial membrane. We're linking movement of electrons and ATP synthesis to its proton gradient. Now remember, whenever you have a gradient, the system will want to reach equilibrium, wants to go back to its normal state. So that's because the proton, think of it like a tiny battery.
我终于知道我电脑是哪里的问题了。
So, what is the ATP synthase? And in simple terms, how does it work? So, this is the enzyme complex catalyzes the conversion of ADP into ATP. Very simple terms. There are two components, the F0 and the F1 subunit. F0 is in the membrane and the F1 protrudes into the mitochondrial matrix. And so, when you look at your notes, this is very simplistic, but when you look at your notes, remember that there is the movement of the protons causes the rotation of the ATP synthase. That rotation allows it to take in phosphate and ADP, joins the two together and spits out ATP. And so we now have that generation of ATP occurring. This is occurring at a phenomenal rate. Remember, you've got multiple ATP synthases that are studied throughout the mitochondria. And the last part is talking about uncoupling of ATP synthesis and why is
很好很好。但我这把必须带你跑。能打呀，能包的。9点卡，9点卡。
achieved. So remember, what we've done to this point is just to make ATP, and that's normal. Why would we want to disconnect it? And that's what uncoupling is. Uncoupling refers to the process of stopping or disconnecting ATP synthesis. And the net result is that it keeps going, but rather than making ATP, we generate heat. And in your notes, I've given the example of animals that undergo hibernation. They need to generate heat. And so one way of doing that is to physically uncouple the ATP synthase, generate heat, and then recouple it so that they're making ATP once they've reached a certain temperature. And for this to occur, there is a protein within the cell, uncoupling protein 1, that is important for this process. It's a normal physiological event. So, energy at the proton gradient normally captured by the ATPase is released as heat as the protons flow through the uncoupling protein 1. and so it short circuits the battery. And if you've ever done this with a real battery, you notice that the battery heats up. You short circuit it, it gets warmer and can explode. But obviously within a cell, that heat is used to heat up the cell, the organ, and in turn the organism. So this is a perfectly normal biological approach. What I want to finish off on though is that we can also do the same thing through chemicals or other molecules that exist in nature. So I gave you several examples. Rotenone is one, which is a fish poison. It doesn't completely block ATP synthesis because electrons can still enter a complex 2, but it's going to reduce that amount. So it has some effect, and obviously as a poison, this is going to be damaging to the individual. You've got an antibiotic, antimycin, so streptomyces can use this as a way of interfering. Remember, ATP synthesis is important for all organisms. ATP is a universal energy source. So if you interfere with its production, that's going to be an effective way to kill off an organism. So antibiotics are going to be perfect for that. You've got other molecules like cyanide, azide, carbon monoxide. These will all inhibit the electron transport chain, ATP synthesis, because they interfere with the ion molecules. And we've just spoken about the iron-sulfur complexes. We said that these were important because they're important in transferring of electrons. So if you interfere with that, then you block the electron transfer, and hence you're going to terminate the whole ATP synthesis process. The last one is dinitrophenol. This was used as a weight loss agent. It uncouples phosphorylation, and so people were using this to generate metabolic energy, converting it to heat, and therefore losing weight. But as you've seen with rotenone and other compounds, and we all know that cyanide is going to be toxic, dinitrophenol is also toxic. And people can actually die, have died, from taking.
this lecture, we're going to cover a new pathway which is called the pentose phosphate pathway, called PPP. And here we're going to look at the importance of ADPH, its generation and its use within the cell. So really quite important, particularly when we come to talk about oxidative stress. So the study aims for this lecture are first of all to understand the two products that are generated from the pentose phosphate pathway. You need to be able to describe the role of ADPH in the cell and understand the pentose phosphate pathway and the key regulatory enzyme, key regulatory step. Understand what oxidative stress is and the role that glutathione plays in this process. And then we're going to talk about this molecule here, pamaquine, that's also called primaquine, how this works, and then we'll also talk about glucose 6-phosphate deficiency and how it can lead to disease. So you've got your section here for the lecture notes in the textbook. So if we start off with our introduction, we know that to date, so far we've been focusing on the breaking down of glucose for energy and glycogen for energy storage. That's been all of the lecture content. But what you need to appreciate is that there is another fate for glucose 6-phosphate. And this one is it's acting as a substrate for the pentose phosphate pathway, or PPP. Now, why is this important? This is the pathway that catalyzes the interconversion of 3 and 6 carbon intermediates, which are end results of glycolysis, with 5 carbon carbohydrates. And these are important steps. These conversions are important because they allow for the generation of pentose sugars. So remember, this is the pentose phosphate pathway, so we're generating a pentose sugar. Pentose refers to 5. These sugars are required for RNA and DNA synthesis, as well as metabolism of 5 carbon sugars that are originate from the diet. But really the key point is this one here. We'll talk about RNA and DNA in a later stage, but for this lecture, pentose phosphate pathway is an important source of the carrier, NADPH. Now we have covered NADH, and we've seen NADPH. Let's just talk about these separately. So NADH is oxidized by the respiratory chain to generate ATP. We saw that in the previous lecture where it was fed in. Depending on where it came into the pathway, we generated different amounts of ATP. NADPH serves as a reductant, so it's a reducing agent for biosynthetic processes. So this is where we want to synthesize something within the cell. But importantly, and it protects against oxidative stress, but it is via this molecule called glutathione. So this is an important distinction to make. So So here we're saying that it protects against oxidative stress, but the actual oxidative stress is dealt with by glutathione, but NADPH has a critical role in maintaining glutathione in its active state. So that's a very easy mistake and a common mistake for students to make. Now when we look at the pathway, there are two phases, and now we have covered NADH, and we've seen NADPH. Let's just talk about these separately. So NADH is oxidized by the respiratory chain to generate ATP. And we saw that in the previous lecture where it was fed in, depending on where it came into the pathway, we generated different amounts of ATP. NADPH serves as a reductant, so it's a reducing agent for biosynthetic processes. So this is where we want to synthesize something within the cell. But importantly, and it protects against oxidative stress, but it is via this molecule called glutathione. So this is an important distinction to make. So here we're saying that it protects against oxidative stress, but the actual oxidative stress is dealt with via glutathione, but NADPH has a critical role in maintaining glutathione in its active state. So that's a very easy mistake and a common mistake for students to make. Now when we look at the pathway, there are two phases, and basically this leads to the generation of NADPH, which is our key molecule, and as we mentioned above, 5-carbon sugars. Now in plants, we're not really going to focus too much, but we know that NADPH is produced by photosynthesis. What about for other organisms? As it shows you again in bold, important step here, NADPH is needed for reductive biosynthesis in all organisms. So that's an important point, part of the synthetic process. And it is also a host of biochemical reactions. And you probably already predict this based on what you know about NADH. NADPH is very similar. Now, if we look at the biosynthesis steps, so these are, if you look at this table, examples of pathways that require NADPH. And as it says here, the red oval will be the focus of this lecture, so this is what we're going to be looking at. But just look here, we've got fatty acid biosynthesis, we've got cholesterol biosynthesis, we've got neurotransmitter and nucleotide biosynthesis. This is all part of that biosynthetic pathway, synthesis of these molecules. Clearly, this is going to be very important. There's also the detoxification. If we go to the second one first, cytochrome P450, this is involved in dealing with compounds by the liver. So these are a series of enzymes that basically break down things that are internalized or ingested by the host, such as drugs, things that need to be metabolized and cleared. But the one that we're going to be looking at is the reduction of oxidized glutathione. So remember, glutathione deals with oxidative stress, and it is oxidized, and therefore the opposite of oxidation is reduction. This is where the comes into play. It allows for the oxidized glutathione to exist in two different states. That is an important concept, an important idea. So if we look at the pathway, as we mentioned above, two phases. One is the oxidative generation of NADPH, and the other one is the non-oxidative interconversion of sugars. This is really where we're going to be interested in this lecture. We'll talk about both, but this is going to be the focus. So in the first phase, we've got our old friend glucose 6-phosphate. It's oxidized, but it's oxidized to ribulose 5-phosphate. You might think about that molecule, think, I've heard of something which is similar. Ribose. So remember, ribose for RNA, deoxyribose for DNA. You can see that this is going to be a precursor. And I've got that on the next line. Ribulose 5-phosphate is then converted to one molecule of ribose 5-phosphate, which is a component of RNA, DNA, ATP, NADH, FAD, and coenzyme A. Super critical component of not only nucleic acids, but you can see all of these other molecules. And when you look at the structure of these other molecules, you will have seen this five-ring structure. So you can see the overlap. But importantly here, two molecules of NADPH are generated. So we're able to regenerate these molecules. And what's the role of the NADPH? Well, it's that oxidative effect that it can have with the glutathione. So the reaction is very simple. As we can see here, glucose 6-phosphate. Here is our phosphate molecule on the 6th position as always. We want to remove protons, so we're going to use glucose 6-phosphate dehydrogenase. NADP plus goes to NADPH and CO2, so it's also a decarboxylation type reaction. So, as it says here, phase 1. For each molecule of glucose 6-phosphate, one molecule of ribose 5-phosphate and 2 molecules of NADPH are generated.
就幾多啊？俾我揾高先過嚟啊！上980個人，我自己看的話980。他把那个人，他把狗给打了，我跟你讲。这玩意儿都被狗给撞坏了。这个女的还没来过。这很大，像有点像。我不愿咬我了，完蛋，我这也没地方躲呀。还有一个地方。
This is going to be
给他让他到，不要过来啊！要是陷入看到这么大的八人
the
角，
rate limiting step,
挺瘆人的。
because this is the one that controls the generation of the molecule that we are interested in. So this will always be the point of control. And if you look here, the Km for NADP plus is 1,000 times greater than for NAD plus. So this is the preferred substrate, and that is important because we need a source of NADPH within the cell. Now, what about that conversion? Well, remember, it's just an isomerization. So ribulose 5-phosphate goes to ribose. We've got just a reorganization of where the molecules are slightly change in shape, or slight change in shape, hence the isomerase. So we've got the same thing, but we've now generated this ribose 5-phosphate, which remember went to RNA, DNA, ATP, etc., etc. So super critical molecule within the body.
但没看到他忘了是干嘛
Now, what about phase 2? What is the fate of the ribose 5-phosphate in cells? But not only we know what the fate is originally, because we're saying, well, it's going to be a substrate or a precursor to here, but what happens when it's in excess to biochemical needs, or we make too much of it?
想个
So ribose 5-phosphate is converted into glyceraldehyde 3-phosphate and fructose 6-phosphate by transketolase or transaldolase. So what does this mean? We've heard of these two enzymes before. These enzymes create a reversible link between the pentose phosphate pathway and glycolysis. So if we have excess ribose 5-phosphate, it can be converted into glycolytic intermediates. So the forward reaction and the reverse reaction can occur here. So this is an important idea.
啊，好像走掉了。
Now, when we look at these tissues with an active pentose phosphate pathway, you can see the normal tissues, but let's focus just on the last one, red blood cells. And if you look here, these are all synthesis. So why am I going to the last one? Because this is the only one where there is no synthetic process, because you know that the red blood cell is there at its terminal stage, but the role is the maintenance of reduced glutathione. And this is what we are going to focus on. Don't lose sight of these above, but this will always be the main focus of this lecture, is the role of glutathione and the role of NADPH in its conversion.
没必要吃了。
Okay, I'll stop there because I want to move on to talk about something else with oxidative stress. So I'll see you in the next recording.
关于预期，我在桌子上休息了。结果一下，
So we finished off by talking about the focus being on red blood cells, and now we're going to move on to the key enzyme glucose phosphate dehydrogenase and oxidative stress. So what is oxidative stress first of all? Now, reactive oxygen species or ROS roles. These are normal byproducts of oxidative metabolism. You know that oxygen is involved in metabolism. We've covered this at length. It carries the electrons as part of the process. But when you use oxygen, you can also generate these reactive oxygen intermediates known as these ROS products. And these molecules can cause damage to all classes of macromolecules if they are not dealt with. So you can have a singlet oxygen, you can have hydroxyl groups, you can have hydrogen peroxide, you can have hypochlorite being generated.
刚才
All of these are very, very chemically reactive and then must be dealt with. Otherwise, you start to have damage to proteins, lipid, carbohydrate, nucleic acids. So how do we deal with these products? This is where glutathione comes into play. Cells use reduced glutathione. So important, it has to be in the reduced state. And they use it as the reducing agent. So what does that mean? Well, if it is the reducing agent, it undergoes oxidation, and it undergoes an oxidation in place of the vital cellular component. So instead of your nucleic acid, this takes the oxidative load. So, glutathione, which is also shown as GSH, combats oxidative stress by reducing reactive oxygen species to water, to non-reactive byproducts, making them safe. Now, what is glutathione? It's just a tripeptide. It is 3 amino acids, and the terminal one has a free sulfur-hydryl group, an SH group. And so you know that this is going to be a cysteine molecule. Cysteine is the only amino acid with an SH group. Why is the SH group important? The SH group allows one molecule of glutathione to bond to another sulfur-hydryl group, forming what is called a disulfide bond. And so because we've got two joined together, this is referred to as a dimer. Now here is the structure of glutathione, and this is shown in the reduced state. So remember, it's ready to be oxidized by, let's just say, hypochlorite or hydrogen peroxide, both reactive intermediates. So here are glycine, cysteine, and glutamate. You can see the sulfur-hydryl group that is here, and you can also then imagine another group that comes along, and we can form the dimeric state. And notice we've got the same directionality that we spoke about previously, carboxyl groups and amine groups on the molecule itself. Now, a little bit of a history lesson. If we now go back to 1926, there was an anti-malarial drug called Camoquin or Primaquin, so these are used interchangeably. People that were given this drug, in certain cases, they were found to undergo hemolysis in these patients, and this is lysis of red blood cells. So this is where they have a breakdown of the red blood cells that's not due to the malaria, but it's actually due to the drug, and this is obviously quite a damaging side effect and could actually cause death to these individuals who are already sick. It took another 30 years to figure out what was going on. And what was identified was recognized that the cause of the drug-induced hemolysis was actually the deficiency of glucose 6-phosphate dehydrogenase. Okay, so this is that key enzyme. Remember, this was the key enzyme rate limiting of NADPH production. So that's an important point. So this is important. But why? As I said here, why is this important? The pentose phosphate pathway is the only source of NADPH in red blood cells. Remember, we keep focusing on red blood cells. That's one where it's not used for synthesis, but it's only used to deal with oxidation.
It's supposed to be the piano job. Eh? 不对吧？谁那么高？他们扔威尔白了。
Producing glutathione. So, if it's the only source, these cells are going to be dependent on its production, and this therefore requires a functional glucose 6-phosphate dehydrogenase enzyme. Now, functional or a present enzyme. So I've said here a deficiency. Now, that could either be an enzyme that is non-functional, or it may just not be present, expressed at very low levels. So there is a problem here. But why is this problematic? Why do the red blood cells need this functional glucose 6-phosphate dehydrogenase? The major role of NADPH in red blood cells is to reduce the disulfide form of oxidized glutathione to the sulfurhydryl form. And it is written this way, GSSG. This has shown you a disulfide bond. That's why it's shown in that particular configuration. Here, the GSH, this is shown as the monomeric form. So it exists in these two alternate states. So what we see, similar to what I just said, we have two monomers in the reduced form. We have our reactive oxygen species. These come together, they're oxidized. And remember, we've got this free proton on one molecule, but we've got two of them, so there's two protons. This leads to water being generated, and whatever has been oxidized is converted to an alcohol, or a much less reactive molecule. But then what happens? Is this the terminal reaction? Well, no, it's not. If we now look to the second step, and this is really the important part, the oxidized form interacts with NADPH. The NADPH is going to donate its protons back to the oxidized form, and we regenerate the monomer. The NADPH goes back to NADP+, and this now needs to be regenerated to make NADPH, and the cycle continues. So the net result is that we have regenerated the reduced glutathione and the monomeric form. And so why is that important? Because this is going to be present, the reactive oxygen species. And so if there is more reactive oxygen species than NADPH, this is going to start to attack different tissues. And if we're looking at the red blood cell, it's going to attack the hemoglobin, and this is what leads to hemolysis. So as it says here, NADPH is thus essential for this reaction. You can see it here. It is critical for that interconversion. If this is not present, then we just go into the forward reaction and we just end up in the oxidized state. And then the excess reactive oxygen species are not dealt with. It's a very, very damaging process. Okay, so just finish off on this example here. Here is our oxidized glutathione. Remember I said GSSG. This is showing you the sulfurhydryl group here, the double bond. Not double bond, sorry, the two sulfurs that are joined together. The NADPH through a glutathione reductase. So remember, this is going to reduce the glutathione and the protons are going to be donated. We end up with two of these because these are split down the middle, and we regenerate this sulfurhydryl group and our NADP+. So you can see that this is able to exist in the reduced versus oxidized state, and this is important for dealing with our reactive oxygen species. Okay, I'll stop there. We'll come back to disease and see why this is problematic. Okay, welcome back. So, what's the mechanism and how does this link to disease? Well, we know that glutathione, so the reduced form, is essential for maintaining the normal structure of the red blood cells because these are dependent on NADPH. If you've got cells with a low GSH or glutathione levels, they're now going to be susceptible to drug-induced hemolysis. I'm not going to go, okay, but why is that the case? Because there is limited levels of reduced GSH to deal with reactive oxygen species. That's really the crux of the matter. So, okay, well that makes sense. So how does this link with disease? Well, when this was first being identified, we now know that up to 11% of African Americans actually have a deficiency in glucose-6-phosphate dehydrogenase. It's an inherited characteristic for this group of individuals. Now, is this problematic? Well, in the absence of oxidative stress, people that have a deficiency in glucose-6-phosphate dehydrogenase are normal. There's no effects, there's no biological consequences, no hemolysis, so it doesn't present. But what we see is that when Pamequin or Primaquin, remember the same name, just two different names for the same thing, this in turn leads to very high levels of oxidative stress. It stimulates oxidative stress. Now, notice this bit that is in yellow. I've highlighted this because this is a very common mistake. Students will think that Pamequin or Primaquin is an inhibitor of glucose-6-phosphate. It is not an inhibitor. It is a molecule that was used to treat malaria, and one of the consequences dealing with malaria was this oxidative stress. Do not make this mistake in logic and understanding. It is not an inhibitor. Make this very, very clear in your understanding. So, how does this lead to disease? So, if we have someone that has this deficiency, NADPH is not going to be generated because remember, glucose-6-phosphate dehydrogenase was the control point and the rate-limiting enzyme, and we've got a high level of oxidative stress. All of the monomeric GSH is going to be oxidized by the reactive oxygen species. We don't have NADPH present, and so we can't return it back to its reduced state. But the level of oxidative stress, the reactive oxygen species, continues to go up because of the drug treatment. Now, reactive oxygen species are being produced. They have to interact, they're reactive, so what are they going to do? They're going to attack the most common protein in the red blood cell, which is going to be the hemoglobin. And by doing so, it damages the cell, damages the protein. Now, red blood cells also lack mitochondria, so there's no other source of NADPH. They're in trouble. They're dependent on this single source. And so what this leads to is lysis, which is what we saw above, the hemolysis that occurs. Now, you might be thinking, well, why did this population of individuals, why do 11% of people have a deficiency in glucose-6-phosphate dehydrogenase? It's actually an evolutionary advantage. It was found to protect against malarial infection. So the treatment is actually the cause of the consequence. So this was an unfortunate combination, as you can see here, a combination of hereditary and environmental factors that led to disease. People were trying to do the right thing, trying to treat people with a drug that could prevent malaria, but actually these individuals were already intrinsically protected against malaria because of this deficiency. What you may not know is that malaria actually infects the red blood cells. So by having this deficiency, it interferes with that replication within the cell and in turn stops the individual from contracting the parasite. Okay, what do we see? Here's an example of red blood cells that have been damaged by a reactive oxygen species. You can see these green circles relating to the hemoglobin that has aggregated. These are referred to as Heinz bodies, and this is evidence of reactive oxygen species interacting with the hemoglobin. And that's because NADPH is not there, the glutathione cannot be regenerated, and we don't have this protective mechanism taking place. So it's very interesting, but also a very powerful demonstration of the role of NADPH and glutathione in dealing with oxidative stress. Okay, that's all for this lecture. So it's a fairly short one, but really quite important. So you can see the chapter summary here. You've got your multiple choice.